I'm going to talk about a simplification of IVF in many respects. The question of why do you want to simplify IVF, because it'll put most of us out of work. And secondly, simplification leads to something like robotics, which is highly applicable in IVF, and so probably in 5 years, these meetings will be quite different. But I got involved in the question of simplifying IVF in a sort of an indirect way. And this had to do with the origin of what we call the simplified culture system. I'll talk a little bit about this, which was published a year ago in RBM Online. And I was asked by Willem Umbelet in Genk, who had been trying for 20 years, quite a remarkable man, to bring art and IVF in particular to low-resource countries, particularly in Africa. And the World Health Organization, through a women's reproductive rights program, was also involved in trying to spread IVF and reproductive technologies in general to parts of the world where it was either inaccessible or unaffordable. And so the challenge that I was asked to be involved in is, could art and IVF be effectively introduced in developing countries where it was much needed, but where resource and infrastructure was limited? And of course, the infrastructure we're talking about is things we take for granted, which is electricity, the constant clean electric source, gases, reagents, etc. And the way they had approached this initially was to ask IVF programs in developed countries, in Europe and the United States, that when they change their equipment, that equipment like incubators, which are all microprocessor controlled, be sort of donated to IVF programs that would be set up in developed countries. And so you essentially duplicate what we have in our laboratories here in the United States in other areas, with the hope that actually you might have a resource poor situation to become a sort of an island of resource rich IVF. Well, it doesn't work. Lots of problems. I'm not going to go into this that was found, but you just can't duplicate what we have in high complexity labs elsewhere. And so the challenge was, how can we do this in a way that might be effective? And the primary goals of this program were to simplify IVF in a way that it was adaptable to IVF settings, adaptable to low resource settings. And it had to be accessible and affordable, which was a real issue because IVF in developed countries is accessible to everybody, or very large numbers of people. But in fact, in most places where you have fairly high infertility rates resulting from sexually transmitted disease, mostly tubal factor, or suboptimal sperm conditions, parameters, it's not available. And there are sociological reasons. There's a whole literature on the consequences of infertility or involuntary childlessness in these areas, which dictated both to people involved in setting up a program that would bring art to these areas and for the WHO to try to have a means by which this could be adapted to, IVF could be adapted in other settings. You also want to reduce the costs to start up a program. It's very expensive, as we all know, to start up a program in a high resource setting. For me, it was to reduce the potential for iatrogenic errors and reduce the need for highly trained laboratory personnel, which sounds counterintuitive, except if you could design a system which was similar to when you buy a printer or a computer and it has a setup, right? You open up the page and there are 5 steps, right, to get the printer or the computer to work. And could that be done in a way that did not require PhDs and did not require very highly trained individuals, but those who had some clinical background and experience? And could you use it in a minimal laboratory infrastructure that is consistent with successful outcomes? And it's important that those outcomes be similar to those in high resource settings, which is asking a lot. So for example, we use medical grade gases, gases that are mixed, pre-mixed at certain concentrations, or just regular gases, and those are not readily available all the time. Same with electricity. Electricity goes on and off in many countries, and so if you're using an incubator, that could be a problem. It could be a problem because voltage changes can fry, and that was one of the consequences of having donated high complexity incubators sent to these countries, is that even with filters, the microprocessor would be wiped out. So these were really difficult challenges, and address this in a way based on the following sort of general principle, and that is that what we know about what human oocytes and embryos need to fertilize and develop in vitro after nearly 50 years, if you go back to the first IVF that was done, not for embryos going back, but just to do IVF in human, we actually know a great deal, more if you include other species, like the mouse, for example. So we know a lot about what embryos need and how they need to develop and under what conditions, especially in the human. And now, of course, in the last 10 years, we could recognize factors that determine developmental competence that are largely uncontrollable, for example, aneuploidy. So we know what human embryos have as intrinsic problems, we know what extrinsic factors they need. So with this knowledge, can IVF be reduced to its essentials, which in reality, taking all the fluff away, is media, pH, temperature, and for embryos to be largely left undisturbed. And so I went to a system that I had used as a graduate student, and that was we were transporting oocytes and embryos over fairly large distances in species that were located in another state. And sometimes it took half a day or more to bring them from Kansas and from Montana to Colorado, where I was working, still am. And the way that we addressed this was an interesting way. It was actually not terribly original, was to have a tube, that generated CO2, and you had a lot of ways you can do this. This was simply using a weak base and a weak acid. We used these sealed vacutainers at the time and had a whole series of these tubes that had CO2 in them. And we would have embryos or oocytes growing, well, some from buffalo, growing in tubes, and we'd be transporting them. And as the media got purple, it was getting alkaline, we just connected a needle with a tube into this, put CO2 in, and then pH went back to normal. So we'd keep doing this over a period of time. And when the embryos came back to the lab and we did analyses on them at the time, the conditions were appropriate for them to continue cleavage and even to form blastocysts depending upon the stage. So I said, well, what if we go back to the essentials and have a system that essentially is really inexpensive to develop? And what it is in this case, we're two tubes that we use vacutainers here, a CO2 generator here, in this case is citric acid and sodium bicarbonate, which is commonly used to generate CO2. It's one of the simplest reactions in chemistry. And you put these guys together, connect them overnight, and the pH, because the CO2 rises, goes down. And so here's what you start with, a pH about 8.3, 8.4, and you wind up with something that's about 7.3. And it's very consistent, remarkably consistent. And what's really nice about this is once you withdraw this tube overnight and take it the next day, these tubes last forever. This atmosphere is sealed. And we've had these now for several years. And I made a whole bunch of these, and once in a while I'll go take it and measure pH, and it hasn't changed after 6 years. So this is a sealed system. It's an enclosed system. And then to do this right, you really want to do quantification. And so we did this is one of the first ones. You have several of these. And you do this by putting a microelectrode for CO2, oxygen, and pH in the tube. And as the CO2 is going in, you measure, take points, and you can have a recording electrode, and you just record it. And you can see the CO2 goes up, and then it sort of stabilizes at about 6%, 5.5%. In this case, oxygen goes to about 12%, and the pH remains constant here at about 7.3, where is that? about 7.4 here. So the point is that you can generate a stable atmosphere, stable environment, and it lasts. It doesn't go away. So you can make these tubes in advance, put them in the refrigerator, take them out, warm them up, and they're still the same. And by changing some of the parameters of time, for example, we're able to get not very different, but we're able to get levels that now are very consistent at 7.3 for pH. We brought the oxygen down to about 5.5 to 6%, as another example. And so this is one that we can sort of change in an interesting way, suitable for different species, especially in this case for the human. And the paper that this was published in, if you want to look at all the details, because there are quite a few details, was in RBL Online in 2014. So the simple thing to do is you now have eggs and sperm, you inject them into the tube, the egg cumulus masses go down to the bottom, you inject the sperm go down to the bottom, you shake it up, put it away. We use about 1,000 to 500 to 1,000 sperm per tube, and it works very well. The fertilization rates, as we showed in this paper and elsewhere, are comparable, I shouldn't say similar, they're the same, if not better than what we're getting in open systems such as a culture dish or even in microdroplets. The only change I've made from this paper is that we use a much longer needle, stainless steel needle that goes all the way to the bottom, and this is useful for denuding the eggs. For example, you can use a little gas-tight pipe syringe, and you can pick up the cumulus masses individually, and just as you would be stripping them in vitro in a conventional way, you can strip them here, and the eggs are denuded. Sometimes it's nice that you leave a little bit of cumulus because they'll stick to the side, but also it allows you to pick up pieces of the cumulus so this media is clear when I show you what we're going to do with it next. So this has made a substantial, simple change, simple-minded change, but actually has been an important one. And then the question is, well, does this work? And the answer is yes, and this just shows the system that you can look at in a conventional hood with a light source underneath. These are embryos that we produce. These are human embryos that were taken out of the vacutator prior to transfer and photographed. This is a picture of hatching. You can see it's not terribly good. It's kind of obscure. I'll show you how it gets better in a minute. And then these embryos were taken out and also transferred. You can see hatching going on here and here. These are not photographed through the tube, but rather through a culture dish. These were, in fact, photographed through the tube. So you can, in fact, get decent imaging, at least through the cleavage stages and the pronuclear stages. And this was the first baby that was produced by this method a couple of years ago. So, to date, it's a small program, we've had, I believe, 78 babies born, all from single embryo transfers, all on day 3, all full term, and no abnormalities so far whatsoever. And the pregnancy rates are about 40% for women who have undergone several IVF cycles and have failed. So this is a closed system that is well defined. And so this is a system that is now going to be used probably in 6 programs by next year. It's low cost. The cost of the tubes, the medium, everything else is about $2. And, there's more, you don't have to use an incubator. You can use a thermos, you can use a water bath, and we now use what are called thermal beads, which are terrific because you heat these beads up, they're stainless steel, they retain temperature for about 24 to 36 hours. You can sterilize them, you stick the tubes underneath, it's dark, and then you take them out. And so these thermal beads have made actually quite a difference. Okay, well that's that, and that's nice, it works. People want to use it, but wait, there's more. Okay. The addition of a fully functional, low-cost, simplified time-lapse and video streaming capacity. Now, time-lapse is a big deal, right? Now, what's nice about this, there was a great comment made this morning about techniques that's the next great thing that don't pan out. And in a sense, time-lapse is maybe, depending upon who you talk to, the next great thing that may or may not be terribly useful if you have good embryology and if you have people who know what they're looking at. But if you're looking at an event that seems to be important, that is occurring at 2 in the morning, it's not going to be very easy to have somebody come back and look at embryos. So time-lapse does, and I'll show you, have utility. So how can you introduce a time-lapse system in a low-cost setting, because the time-lapse machines are very expensive, and the material that you have to put the embryos into is very expensive. So it adds a lot of cost, and so the low-cost system is gone. So I've been working on this for quite some time, and I thought, what can I get for free? And the reason I like that is my son is amazing about finding freeware, okay, that is fairly useful, some of which is probably shouldn't have been freeware. But in any event, there's a whole generation of people who can find programs and everything else that is quite nice to use and free, more or less. So I'm going to show you how we do this. This is our time-lapse system. And it's not only time-lapse, but it's video streaming. And if everything works, I'll show you an embryo that we have now that is going on in the lab in Boulder. So what we use is a microscope, right? And you can fairly inexpensive one, not phase, not Nemarsky, it is just bright field optics, very inexpensive lenses. This is the lights. We've used Olympus. There's some really nice, inexpensive inverted microscopes from China that are also very useful. This is a tube. You'll see this in a little bit more in a minute. The embryos are going to be in here. This is a heater, $9.95, right? And this is an old way that people used to do this type of stuff years ago. This is nothing new. You have a heater, and it forms an air curtain, right? And you define the distance between the heat and the BTUs that are coming out, turn it on, and you can show it, I'll show you in a minute, that you can get a stable 37-degree temperature here. Very easy. We use a computer which has a program in it, the one we're going to use, I'll talk about again, which was found on the internet. It's called the EvoCam. Most of these programs, incidentally, are surveillance programs for convenience stores, right? For preventing thieves, etc. Okay. We use just in Colorado, because we've had some problems with changes in construction on the campus, and there's been interruptions in voltage and voltage drops, etc. So we have added a line conditioner and an automatic voltage regulator, very inexpensive, to get a clean signal. And this is just a time date generator. Okay, so this is what we stream through this program, and this is going to be watched anywhere in the world. It's also recording, so you have archival time-lapse as well. And so this is the image that we see through the microscope, and this is what's going out. Now, the interesting thing about it is that the lights are on here. You turn the lights off, you can't see anything. There's no light, virtually no light coming through. And what we use is a 633 nanometer infrared filter that's in the optical path, because this increases the contrast. And so it's monochromatic. We get one frequency, which is, I think, safe, supposedly, for eggs, but it also increases the contrast significantly. Okay. Now, this is an Eclipse microscope, Nikon, and what you're seeing here, I hope you can see it, is that we have a thermocouple here. This is where the embryos would be. It's lying flat on the scope, and this is saying 37.2 because that's where it's measuring right here. There's a conventional thermometer over here, and you can see it's measuring 37. And that's stable forever, as long as the electricity is on. And you can see the tube that's right here. There's a vacutainer, and we elevate it by about 10 degrees using modeling clay. So here's the lens. It's elevated this way, and the embryos are here. And we can see up to 6 embryos at a time. Okay? This whole setup if you do it right, is about $800. And that is including the microscope. The camera that we use is a analog camera, which is used in surveillance. It's not terrific resolution, but it's actually quite good. There are a number of manufacturers like Panasonic, et cetera, who make these. I think I have one here. It's Panasonic. Since it's analog and this is digital, for processing, you have to get an inexpensive converter, which converts the analog to a digital signal. So you save hundreds and hundreds of dollars because you don't need color. You just need a monochromatic camera, CCD, or, well, this is CCD. Okay. All right. So what do we get? All of these were taken through the tubes. This is normal pronuclear formation. This is the time. And you can just go through the sequences, and you can see pronuclei forming here, 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 and then disappearing. Okay? So these were taken from the video stream. So we had people all over the world watching this. In fact, this will be the first, I hope, if it works, to see one that's in real life. So this is good. It's good for a number of reasons. You can assess this. The image that you're seeing here, by the way, is we've measured the light. It's under 0.05 lux, which is virtually nothing. Yet you still get a very good signal through this. The program we use is called EvoCam. It costs about $10 for a license. It's actually free for universities, and it is a fully functional, multifunctional program. So these exist online. You don't have to, you can find them if you know where to look. I don't know where. My son knows where. You can find anything. Okay. Where is it useful? Well, abnormal first cleavage. In this case, what you're seeing is pronuclear formation, first cell division, bum, 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 syngamy, first cell division to 3 cells. That's been described. Okay? Well, it's important because if you come back and look at this embryo a little later, 38 hours, this is a normal-looking embryo. You wouldn't know that this has happened. In fact, if you looked at this embryo by its genetics, it is a mosaic. It's chromosomally mosaic. So this is one useful function for this, is that you can see abnormalities in cleavage. Not so much the normal stuff, but it's the abnormalities. Okay, here's another one, abnormal second cleavage, which is actually relatively recent but not unusual. Cells divide. This again is through the tube, no undisturbed, dark room. First cell division gives you 2 cells. Then all of a sudden, you get 3 cells from one blastomere. 1, 2, 3. Goes through again. You have 1, 2, 3. You have 5 cells. There's another cell on the other side. And as this goes along, it's not too bad as of an embryo. And if you didn't look at these stages, again, you would have missed this. Here's blastocyst formation beginning in this particular embryo. So that we can see. Okay. Normal blastocyst hatching. What's nice about this is it shows you the resolution that you can get. Here's a cell, here's the cell dividing in the trophectoderm. Right? This is hatching. And I'll show you the video at the time lapse of these in a minute. So here are the stages where the embryo is emerging, etc. Again, stages that you can see in a system. It's an open incubation system. It's a closed culture system, but an open incubation system. Failed hatching. Embryo is going to try to hatch. Here are the zone of breakers that it's going through. It doesn't do it. It expands, collapses, and then stops. And I'll show you the time lapse of this in a minute. Now, this is where it gets really interesting. I mean, really interesting for me. You leave it in culture, right? It's a hatched embryo. Just leave it. This is culture media. It's just the same sort of Global T or Irvine, doesn't make a difference. And we watch these guys, and after about 65 hours post-hatching, the embryo collapses, and we start seeing these cells coming out. Okay? And you can see them move in the time lapse. This is right through the tube. 175 hours, we have this really bizarre structure with sort of this material, whatever it is, over here. So we thought, this is like embryoid bodies. These are like ES cells. So to show that, what we found is that, in fact, these are HES markers that are positive here. In this particular structure, when we look for OCT3/4, it's intense here, and right about here, it gets less intense, but it's still there, and it's sort of, this has been amplified a little bit. This is taken through the tube, in situ. Okay? And so the OCT3/4 is a really good measure of inner cell mass derivatives. Seed there, and the region here, which is OCT3/4 positive, is CDX to weak, I'm going to say minor. And at 185 hours, we start looking at this region right here where these cells were coming out, and we see this sort of very strange matrix. The matrix turns out, when you look at it, to be neurites. And what we stained with a variety of markers for different cell types, and the one that was positive is the one for nestin, which is an intermediate filament protein that is characteristic of early stem cell differentiation into neurons. And you can see these fibrils here that were over here, and then stained, the same one stained with nestin nucleus, and if you imagine like the canals on Mars, you might see this line that is right here. So what we're getting, without any addition growth factors, without any addition of anything, there's the spontaneous formation of what we think is an embryoid body. Conditioned media makes its own factors, probably makes its own matrix to stick to the surface, and early forms of differentiation. So this is a stem cell marker, as I said, for neural cells that we're seeing in this structure that's now outside of the embryoid body. And we have other now cells that are going into differentiation into other cell types. So this says that this is a robust system, not just for making embryos and making babies, but it actually goes beyond that. And by changing the media, by sticking a syringe in, you can put in, which we intend to do, media that will perhaps get embryos to a more later stage of differentiation, but allows you also to take that fluid out that the embryos are around and see what factors are there. And that's the process we're looking at by mass spec, is to see what factors the embryo itself has produced spontaneously. Okay, so clinical applications, training and remote viewing for simultaneous video and voice discussions. You can use Skype for quality control, selection, deselection, research opportunities. Point being is that we have set this, we're going to set these up in some of the programs that have adopted this simplified culture system, and so people around the world could, anyone at any one time, talk to people in, let's say, we have it in Ghana, and say, this is an embryo that you want to look at, these are issues that you're having with development that you may want to address. So you don't have to be there, you can be elsewhere. You can detect cytokinetic abnormalities. I've shown a few that contribute to aneuploidy that may be missed in static observations if occurring at inopportune times, post-insemination. So that's another potential use of it. Now, there's only one embryo per microscope. I mean, so no one's going to set up 16 microscopes. But if you have a patient who has chronically failed IVF or whose embryos have shown abnormalities in development, this is one way where you can select that patient for this type of analysis, even in a high-resource program, and see what may be going on. Basic research, sampling of media for secreted biomarkers that may be viability markers. We're using it to visualize the effect of activators, calcium ionophores, and metabolic inhibitors. In situ, studying molecular and cellular aspects of post-hatching development and putative ESL formation differentiation. And most recently, the effects of CRISPR-Cas gene editing at the earliest stages of embryogenesis to post-hatching, and we want to extend that to day 14, which means that if you can edit the genes, knock out specific genes in an embryo and leave that in culture in situ and do nothing else, you can ask what effect it may have downstream. Okay, I want to end by current directions. I'm going to forget about this guy, and then just say that we've actually done this in the bovine, and we've been able to do in vitro maturation, in vitro fertilization, development of blastocysts in the same culture, and then we take the tubes and they're transported from the laboratory to a field, sometimes about 40 miles away for impregnation, to the animals, and it seems to work pretty well. So this has industrial applications as well. Okay. We now have programs that have been written for auto-contrast and focusing, so that, in fact, the embryos there will be no difference in contrast, and a region that is defined as something we want to look at will be in focus and throughout. Okay. And, oh, sorry. These are the two people who've worked on this, Christine Hennigan and Sarah Zimmerman, who've done a lot of this work. Okay, now if this works, I want to show you it'll take 2 minutes. Okay.
Now, if you would like to collect one question from the audience, you have to choose. You take 2 minutes, or you can collect some comments or questions.
I want to show what you see. Okay, if I can get this to do it. All right, so you have to help me to. Okay. So there should be a bar at the bottom. Yeah, so I'll go with that, okay. Right, it should be. There it goes. This is hatching. The bulb burned out, that's why it went up right. Okay. So this is hatching, and you can see this is right through the tube. Everything left alone. Okay, go to the next one. And that's fine. And if I can get this right, what I want to do is show you a live specimen that's on the, let's see, so I need to go to escape. Escape in this, where's escape? Escape.
Like this?
Yeah, I guess, no, okay, so escape, and I need to go to Firefox, which is right there. Okay. Well, it's on here, but there it is. So I don't know what happened, it sort of moved. So this is now sitting in a lab. This was a 20-year-old frozen embryo, 2-cell, 1-cell lysed, and it's been in culture it's still in culture for 3 days, and you can't see it, but this is the type of imaging that you can send anywhere in the world. In fact, this is probably not the best, but it just shows that this is what we can see. And you can put 4 or 5 embryos on this by lowering the power of the objective lens. Okay, so, yeah. Right.